Welcome to our Bioprocess Engineering online tutorial on industrial bioprocesses. Through the Bioprocess Engineering online tutorial series, you gain expertise in the principles of bioprocess engineering and their applications to enzymatic bioconversions, microbial fermentations, and cell cultures bioprocesses. For more details, please visit us at bioprocessengineeringonline.com. In this tutorial on industrial bioprocesses, we'll address three questions. What are bioprocesses? What are their applications? And what are their constitutive unit operations? Industrial chemical processes consist of sequences of operations, transforming raw materials into bioproducts. In bioprocesses, a central step is a biotransformation, utilizing enzymes or living cells. Some bioprocesses may entail multiple biotransformation steps, such as consecutive enzymatic bioconversions or enzymatic conversion followed by microbial fermentation. Bioprocesses are broadly categorized into four classes. The first category of bioprocesses comprises enzymatic bioconversions. In these processes, an enzyme protein catalyzes the cleavage or recombination of molecules. Enzymes hold industrial significance for their wide range of reaction activities in aqueous and non-aqueous media, and for their mild operation conditions such as temperature and pH. Moreover, their regio and enantioselectivities selectivities make them particularly attractive for synthesizing chiral molecules. The second category of bioprocesses comprises microbial fermentations. On an industrial scale, a wide variety of microorganisms, including bacteria, yeasts, and filamentous fungi, are utilized. In these processes, microbial cells leverage their intricate metabolic pathways to proliferate and yield a broad spectrum of products. These encompass small molecules like ethanol and lactic acid, larger macromolecules such as proteins and polysaccharides, as well as fermented products like beer. Furthermore, these microbial cells can be subject to metabolic engineering, enabling the production of recombinant proteins and novel metabolites. Additionally, microbial cells are also used to degrade and eliminate pollutants. The third category of bioprocesses involves animal or mammalian cell cultures. Animal cells, characterized by their more elaborate metabolic pathways, can be engineered and cultured to synthesize and excrete proteins and glycoproteins. Animal cells find also applications in the propagation of viruses. The fourth category of bioprocesses comprises plant cell cultures. On an industrial scale, the distinctive metabolic capabilities of plant cells are harnessed to generate a diverse range of phytochemicals, including pigments, aromas, flavors, dietary supplements, and biostimulants. Additionally, plant cells can be engineered for the production of recombinant proteins. Enzymatic and cellular bioprocesses find diverse applications across various industrial sectors, including human food and animal feed, human and animal health, commodity and specialty chemicals, energy, particular biofuels, and materials such as biopolymers. Moreover, bioprocesses play a crucial role in waste and water treatments. Bioprocesses play a central role in the area of biorefineries. Biorefineries transform renewable agricultural and forestry resources into a diverse array of bio-based products, including food, feed, chemicals, materials, as well as in biofuels and energy. Biorefineries make use of both enzymatic and microbial processes. They involve the initial conversion of biomass into bio-based intermediates, particularly sugars. Subsequently, these intermediates undergo further transformation into various chemicals and polymers. In the domain of biorefineries, bioprocesses are characterized by very large production scales up to millions of tons, bioreactors from hundreds to several thousands of cubic meters, relatively low product purities, low production costs from less than 1 to a few euros per kilogram, and high environmental constraints. Bioprocesses play also an essential role for the production of biopharmaceuticals. Enzymatic bioconversion processes are increasingly employed for the synthesis of chiral drugs and elaborate molecular structures as mRNA. Microbial fermentation processes have been traditionally used for the production of antibiotics. More recently, fermentations with recombinant bacteria and yeasts were implemented for the production of peptides, proteins, DNA, and viral particles. Today, the majority of pharmaceuticals biotransformation operations makes use of animal cells for the production of glycosylated proteins as monoclonal antibodies, viruses as vaccines or vectors for gene therapy, as well as active cells for cellular therapy. Within the biopharmaceutical domain, bioprocesses are distinguished by their operation on a relatively small kilogram scale, utilizing bioreactors with capacities ranging from hundreds to thousands of liters. The production costs associated with these processes are notably high, spanning from hundreds to millions of euros per kilogram. Stringent adherence to regulatory standards is of utmost importance, with a particular emphasis on ensuring product purity, quality, and consistency. In industrially employed bioprocesses aimed at transforming raw materials into bioproducts, a distinction can be made between the upstream and downstream sections of the process. The upstream section encompasses the pretreatment of raw materials, the preparation and sterilization of media, as well as the enzymatic or cellular biotransformation. On the other hand, the downstream section involves operations for the recovery, purification, and ultimately the formulation or drying of bioproducts. The number of downstream operations is contingent upon the level of product purity to be reached. At the core of bioprocesses is the biotransformation operation. This transformation process involves the conversion of raw materials into bioproducts, a task accomplished through the use of enzymes or living cells within bioreactor equipment characterized by diverse sizes and geometries. Commonly employed unit operations in bioprocesses are thermal processes. These include heat exchange operations for fluid heating or cooling, sterilization operations designed to eliminate microorganisms in fermentation media, evaporation processes aimed at concentrating solutions, and drying techniques such as spray drying or freeze drying for bioproducts. The downstream section of bioprocess comprises separation operations dedicated to the recovery and purification of products. Among these, traditional separation methods from chemical engineering are employed, encompassing precipitation, crystallization, adsorption, ion exchange, and distillation. Furthermore, bioprocess separation operations incorporate more biospecific methods, such as membrane-based purification, which includes microfiltration, ultrafiltration, nanofiltration, reverse osmosis, dialysis, and electrodialysis. Additionally, chromatographic purification techniques play a vital role to reach high purities, encompassing ion exchange, hydrophobic, size exclusion, and affinity chromatography. The final objective of bioprocess engineering is to achieve both economic efficiency and environmental sustainability in bioprocessing. In order to effectively design and optimize bioprocesses, the process engineer must carefully consider the entire sequence of both upstream and downstream operations. A critical aspect involves assessing how the performance of the central biotransformation step influences the sizing and effectiveness of other operations, both upstream and downstream. Moreover, the process engineer can strategically explore the potential for recycling materials, especially water and heat among unit operations. This approach not only contributes to reducing the overall production cost of the bioprocess, but also minimizes environmental impacts associated with resource consumption. In conclusion, in this introductory tutorial on industrial bioprocesses, you learned: what are the bioprocesses involving enzymes, microorganisms, animal and plant cells? What are the major industrial applications of bioprocesses for the production of food, chemicals, and pharmaceuticals? What are their upstream and downstream unit operations which have to be considered when designing and optimizing bioprocesses? If interested, you can gain a deeper insight in industrial bioprocesses and their design by enrolling to the online course, Introduction to Bioprocess Design, Scale-Up and Simulation. For further information, please visit our Bioprocess Engineering online website or contact us at bioprocessdigital.com. We hope you found value in this introductory tutorial on bioprocesses. Your feedback is important to us, so please share your comments with us. Explore our website for more information about our comprehensive online courses in bioprocess engineering, and be sure to subscribe to our Bioprocess Engineering online channel for upcoming tutorials. Thank you for watching.